Of all the challenges we face with climate change, one of the most underrepresented is what it means for our food and water supply. With the continuing buildup of CO2 and other greenhouse gases in the atmosphere, climate scientists predict that there will be more extreme weather events, including more severe and extended droughts in much of the world. This will not only lead to conflict over water as a resource, but could have negative impacts on the food supply if we do not have enough fresh water in regions of agriculture. My name is Shea Moore Farrell, and I am studying drought tolerance in plants in the hopes of metabolically engineering food crops that require less water. My lab uses sorghum, a diverse grain crop similar to corn, to study how plants respond to drought stress. Sorghum is one of the oldest crops in the world, and over time it has adapted to many different environments. For example, some varieties evolved in wet and rainy climates and require much more water than those varieties that evolved in hot and dry climates. This difference in drought tolerance within the same species offers a unique opportunity to study how genetics and metabolism affect the quantity of water a plant needs to remain productive. All plants grow through a process called photosynthesis, where energy from the sun, water from the soil, and carbon from the atmosphere are combined to form biological molecules. Carbon is the key element here, as it makes up all the molecules necessary for plant life to be possible. Some of these molecules store energy, like glucose or starch. Some are hormones responsible for growth and development, like auxins and cytokinins. And some are important for the response to environmental or biological stress, like jasmonic acid. Depending on what the organism is doing at any given time, the rate at which carbon gets added to these different molecules, called metabolites, will change, and this has a direct impact on the organism's physiology. All of this occurs through an intricate and complicated web of chemical reactions called a metabolic network, and the movement of carbon through this network is termed carbon flux. A good analogy for carbon flux is cars moving on roads and highways. When hopping in your car, there are many possible routes to get to your destination, and generally the fastest way is to take highways due to the higher rate of speed. This isn't always the case, however, as we've all experienced hitting traffic on highways due to an accident or construction. This slowdown is often due to lanes being blocked or closed, which forces all remaining cars through a smaller space, or a bottleneck. If you are smart enough to check Google Maps for traffic before leaving, you have the option of taking an alternative route around the bottleneck and saving time on your commute. In a similar fashion, carbon moves through a metabolic network. In a plant's case, it starts as CO2 and then moves along the various reactions to its destination. Carbon could end up in any of the metabolites in the network, and we are interested in determining the rates in which it gets there. Similar to going around a bottleneck on a highway, if there is a bottleneck in the flow of carbon to an important metabolite, we are hoping to figure out ways to direct carbon around it. To help us make sense of flux through the hundreds or thousands of chemical reactions within the network, we use computational models much like a transportation engineer would use models of cars on the road to study traffic patterns. To accurately measure carbon flux in a living organism, we use a technique called metabolic flux analysis. This involves feeding labeled carbon atoms to plants in the form of CO2 and then tracking these atoms to see which metabolites the carbon gets integrated into over a given amount of time. For example, before feeding any labeled carbon, none of the metabolites in the network would contain those atoms. If we fed the plant labeled carbon for 1 minute and then took measurements, we would expect the labeled carbon to only be integrated into the metabolites near the beginning of the network because it wouldn't have had time to move any further. However, if we fed the plant for a longer period of time, say 5 minutes, we would expect the carbon to have made it further down the network because it has had time to pass through more of the reactions and thus reach more of the downstream metabolites. So by measuring the level of metabolite labeling at various time points, we can get an idea of how quickly carbon is moving through the different branches of the network. As was discussed earlier, the rate at which carbon is assimilated into different metabolites is directly influenced by the plant's response to its environment. One of these environmental factors is stress, and drought is a form of stress that radically changes a plant's physiology. From this comes our hypothesis, which is that during drought, carbon flux increases through the metabolic pathways that generate metabolites necessary for the drought response. Additionally, we believe that drought-tolerant varieties reroute carbon much more efficiently than drought-susceptible varieties. To test our hypothesis, we use a common experimental setup called a dry down, which simulates drought in plants by withholding water for 6 days. Then, by performing metabolic flux analysis during the dry down, we can investigate how the flow of carbon differs between drought-tolerant and susceptible varieties. The idea is that this will highlight which specific metabolic reactions and pathways are important for a plant to survive drought. Once we have identified which metabolic pathways are important for drought tolerance, we can target specific reactions and strategize methods for increasing carbon flux. Many strategies exist to accomplish this goal, such as overexpressing the enzyme that catalyzes the reaction with the bottleneck, introducing entirely new pathways to divert carbon around the bottleneck, or relieving chemical feedback inhibitions. By directing more carbon to the important metabolite, we would expect the organism to display improved drought tolerance when compared to natural varieties. If so, this approach could be applied to many important agricultural crops. This would improve freshwater security by reducing a significant drain on the resource while simultaneously allowing these plants to survive the predicted changes in climate and continue to feed our growing population.